From marine waste to advanced biomaterials, Chitosan-based systems for biomedical and materials science applications. Chitosan, a cationic polysaccharide derived from chitin, has emerged as a versatile biomaterial owing to its biodegradability, biocompatibility, antimicrobial activity, and chemical tunability. This video presents a comprehensive review of Chitosan production from marine biomass, including shrimp shells, crustacean waste, and fish scales, with emphasis on extraction methodologies and process optimization. The intrinsic limitations of native Chitosan, particularly poor solubility and structural heterogeneity, are discussed alongside advanced chemical modification strategies designed to enhance its functional performance. Recent developments in Chitosan-based nanomaterials, drug delivery systems, tissue engineering scaffolds, antimicrobial coatings, and sustainable packaging technologies are critically evaluated. The integration of Chitosan into next-generation biomaterials underscores its potential in biomedical and materials science applications, while highlighting challenges associated with standardization and industrial scalability. The growing demand for sustainable, biocompatible materials has intensified research into naturally derived polymers. Among these, Chitosan has gained significant attention due to its unique physicochemical and biological properties. Chitosan is obtained through the partial deacetylation of chitin, one of the most abundant natural polysaccharides, primarily sourced from crustacean shells such as shrimp and crab, as well as from alternative sources including fungal biomass and fish scales. Structurally, Chitosan consists of beta-1,4 linked D-glucosamine units with varying degrees of acetylation, resulting in a polydisperse polymer system rather than a uniform macromolecule. The presence of primary amino groups imparts a cationic character to Chitosan under acidic conditions, enabling interactions with negatively charged biological membranes, macromolecules, and surfaces. This property, combined with film-forming ability and mucoadhesion, makes Chitosan highly attractive for biomedical and industrial applications. However, its limited solubility at neutral and basic pH and variability in molecular weight and composition pose challenges that must be addressed through controlled processing and chemical modification. Materials and methods. Raw materials. Marine biomass, particularly shrimp shell waste and fish scales, such as those derived from Prochilodus magdalenae, serves as an abundant and sustainable feedstock for Chitosan production. These materials are often generated as industrial byproducts and represent an environmental burden if not properly managed. Their utilization for Chitosan extraction contributes to waste valorization and circular bioeconomy initiatives. Extraction of Chitosan. The extraction of Chitosan from marine biomass involves a sequence of chemical treatments designed to isolate and convert chitin into its deacetylated form. Initially, the raw shells or scales are thoroughly washed to remove adhering organic matter, followed by drying and mechanical grinding to obtain a uniform powder. This pretreatment enhances the efficiency of subsequent chemical processes. Demineralization is performed using dilute hydrochloric acid, typically in the range of 1 to 2 normal, at ambient temperature for approximately 24 hours. This step removes inorganic minerals such as calcium carbonate, which are abundant in crustacean shells. Following demineralization, the material is subjected to deproteinization using sodium hydroxide solutions at elevated temperatures, 60 to 80 degrees Celsius. This treatment eliminates residual proteins, yielding purified chitin. The critical step in the process is deacetylation, in which chitin is converted into Chitosan through treatment with concentrated sodium hydroxide solutions. The degree of deacetylation, DDA, achieved during this step determines key properties such as solubility, charge density, and biological activity. The efficiency of the overall process is typically reflected in yields ranging from 5% to 30%, with reported values around 23% for shrimp shell-derived chitosan, consistent with literature reports. Fish scales represent an emerging alternative source of chitosan. In these systems, milder deacetylation conditions can produce low molecular weight chitosan with enhanced antibacterial activity. Such materials have been successfully incorporated into biodegradable polymer films, demonstrating their potential in sustainable packaging applications. Structural and physicochemical properties of chitosan. Spectroscopic analysis confirms the successful conversion of chitin to chitosan with observable changes in characteristic absorption bands corresponding to the loss of acetyl functionalities. Morphological studies reveal that chitosan exhibits a range of structural forms, from fibrous networks in its bulk state to well-defined nanoparticles depending on the synthesis method employed. These structural variations play a critical role in determining functional performance in different applications. Chemical modification of chitosan. To address the limitations associated with native chitosan, extensive research has focused on chemical modification strategies targeting the reactive amino and hydroxyl groups. Quaternization introduces permanent positive charges, significantly improving water solubility and antimicrobial activity. Carboxymethylation results in water-soluble derivatives that are particularly suitable for drug delivery applications, including targeted delivery to organs such as the liver. Other modification approaches, such as acylation and hydroxyalkylation, disrupt intermolecular hydrogen bonding, thereby enhancing solubility and flexibility. Thiolation introduces sulfhydryl groups, improving mucoadhesion and facilitating enhanced permeability across biological membranes. Phosphorylation and sulfation impart bioactive functionalities, including anticoagulant and osteoinductive properties, by mimicking naturally occurring biomolecules. Pegylation further enhances nanoparticle stability, prolongs circulation time in vivo, and reduces immunogenic responses. Preparation of chitosan nanoparticles. The fabrication of chitosan nanoparticles is achieved through several established methodologies, each influencing the size, morphology, and functionality of the resulting materials. Ionic gelation using tripolyphosphate, TPP, relies on electrostatic interactions between the positively charged chitosan chains and negatively charged cross-linking agents, resulting in the formation of nanoscale particles. Alternatively, covalent cross-linking using agents such as glutaraldehyde produces more rigid and porous structures. Polyelectrolyte complexation with polymers such as sodium alginate yields hydrogel-like systems with enhanced stability. The incorporation of therapeutic agents such as salicylic acid during nanoparticle synthesis allows for the development of drug-loaded systems with controlled release properties. These systems are particularly relevant for antimicrobial and antibiofilm applications. Characterization techniques. A combination of analytical techniques is employed to characterize chitosan and its derivatives. Fourier transform infrared spectroscopy, FTIR, is used to confirm the removal of acetyl groups and the presence of characteristic functional groups. Scanning electron microscopy, SEM, provides insights into surface morphology, while transmission electron microscopy, TEM, enables detailed visualization of nanoparticle size and internal structure. Dynamic light scattering, DLS, is used to determine particle size distribution, typically in the range of 80 to 400 nanometers, and zeta potential measurements provide information on colloidal stability, with values above +30 millivolts indicating stable dispersions. nanoparticle morphology, and functional performance. Microscopic analyses demonstrate that nanoparticle morphology is highly dependent on the preparation method. Ionic gelation techniques typically yield small, uniform particles, whereas covalent cross-linking and polyelectrolyte complexation result in larger, network-like structures. Zeta potential measurements indicate strong electrostatic stabilization, which is essential for maintaining dispersion and preventing aggregation. Drug-loaded chitosan nanoparticles exhibit enhanced stability and controlled release characteristics. In the case of salicylic acid-loaded systems, improved antibiofilm activity has been observed, highlighting the potential of these materials in antimicrobial therapies. Biomedical applications. Chitosan-based materials have found widespread applications in the biomedical field. In drug delivery, their ability to encapsulate and protect therapeutic agents enables controlled and targeted release, improving bioavailability and reducing side effects. In tissue engineering, chitosan-based scaffolds support cell adhesion, proliferation, and differentiation, with modified derivatives showing enhanced performance in bone regeneration and osseointegration. The antimicrobial properties of chitosan arise from electrostatic interactions between protonated amino groups and negatively charged microbial cell membranes, leading to membrane disruption and cell death. This mechanism is effective against a broad spectrum of microorganisms, including both gram-positive and gram-negative bacteria. Additionally, chitosan-based wound dressings promote hemostasis, maintain a moist environment, and facilitate tissue regeneration. Materials, science, and sustainable applications. Beyond biomedical applications, chitosan plays a significant role in materials science and sustainability. Its compatibility with other polymers enables the formation of composite materials with enhanced mechanical and barrier properties. Blends with polymers, such as polyvinyl alcohol, polyethylene glycol, and collagen, have been widely explored. Chitosan also serves as a green-reducing and stabilizing agent in the synthesis of metallic nanoparticles, providing environmentally friendly alternatives to conventional chemical methods. In the field of food packaging, chitosan-based coatings offer antimicrobial protection and extend shelf life. Hybrid systems, such as chitosan polydimethylsiloxane coatings, provide water and oil-resistant surfaces while maintaining biodegradability. The use of fish scale-derived chitosan in biodegradable films further highlights the potential of this material in sustainable applications. These films demonstrate effective antibacterial activity and represent a viable alternative to conventional plastic packaging. In conclusion, chitosan represents a highly versatile and sustainable biomaterial with significant potential across biomedical and materials science applications. Its derivation from marine waste not only addresses environmental concerns, but also provides a renewable source for advanced functional materials. While substantial progress has been made in overcoming the limitations of native chitosan through chemical modification and nanostructuring, challenges remain in achieving consistent material properties and scalable production processes. Future research efforts should focus on the development of green extraction techniques, standardized characterization methods, and translational studies to facilitate the integration of chitosan-based materials into commercial and clinical applications.